Hello everyone. In the second part of the lecture, I will discuss about the classification of bacteria again. This is a concise presentation for dental students. This chart shows a brief summary of bacterial classification. You can classify the gram-positive bacteria into cocci or coccobacilli. The cocci bacteria can be subclassified into bacteria that are found in clusters and those that are found in pairs or in chains. The cocci group of gram-positive bacteria that are normally found in clusters are staphylococci, and the staphylococci they show coagulase test positive. So what is the coagulase test? It is a type of test which is used to differentiate coagulase positive Staphylococcus aureus from coagulase negative Staphylococcus aureus. Coagulase is basically an enzyme which is produced by Staphylococcus aureus that converts soluble fibrinogen in plasma to insoluble fibrin. This test is performed in vitro, that is in laboratories. Whereas the cocci, which are found in pairs or chains, normally include Streptococci species and Enterococci species. They show hemolysis test positive and they are also classified according to the Landsfield serotyping. To understand this, you have to first of all know about the hemolysis test and the Landsfield serotyping. I will give a brief introduction to the Landsfield serotyping. Many of the Streptococci can be classified under the Landsfield system. In this case, they are divided into a number of distinct serologic groups on the basis of carbohydrate antigens in their cell wall. These antigenic groups are designated by the letters A through T. The Landsfield serologic groups A, B, C, D, F and G are the ones that normally infect human beings. Not all pathogenic streptococci can be identified by Landsfield typing. Example, Streptococcus pneumoniae. The bacteria Streptococcus pneumoniae cannot be classified according to their Landsfield serotyping. In my next slide, I will discuss about the hemolysis test. Now we come to the gram-negative bacteria. Similar to gram-positive classification, gram-negative bacteria can also be classified into cocci and coccobacilli, but here we have another classification that is according to the structure, that is rod. So gram-negative can be classified into three main categories according to their morphology, that is cocci, which includes examples such as Neisseria and Moraxella. Rod, that is they have a rod shape, they are also known as bacilli. Examples are lactose fermenting rods, example E. coli, Klebsiella and Enterobacter. Examples of lactose non-fermenting rods, examples are Pseudomonas, Proteus and Stenotropophonas. Examples of lactose slow fermenting rods, they are Serratia and Citrobacter. Examples of anaerobic bacteria, we have Clostridium species, Bacteroids, Peptostreptococcus and Actinomyces. Examples of atypical bacteria, it includes Mycoplasma, Chlamydia, Rickettsia and Legionella. Examples of coccobacilli gram-negative rods. They have a structure which is intermediate in between cocci and rod. Example includes Haemophilus bordetella, and examples of gram-variable coccobacilli, that is they can be either gram-positive or gram-negative. Example is Acinetobacter. My next slide is about the hemolysis test. We can classify streptococci into three major categories. They can be either alpha hemolytic bacteria, they can be either beta hemolytic bacteria, or they can be gamma hemolytic bacteria. So the scientist Brown in the year 1919 he introduced three terms, alpha, beta, and gamma, to indicate three types of streptococci, which is based on hemolytic reaction on blood agar plate. So this is the blood agar plate. He did this experiment and classified streptococci into three major categories, that is alpha, beta, and gamma. So what does this mean? First of all, we have alpha hemolytic bacteria. Such bacteria, they show partial or green hemolysis. As you can see in this picture, the alpha hemolytic, they have a partial hemolysis. The area surrounding this is partially hemolyzed, and they show green hemolysis. It is dark green in color. Why this is green? Because there is reduction of hemoglobin. That is, hemoglobin is converted to methemoglobin. Examples of streptococci is Streptococcus pneumoniae. It shows partial or green hemolysis, or also known as alpha hemolytic streptococci. Next we have beta hemolytic bacteria. They are associated with complete lysis of the RBC surrounding the colony. As you can see in this picture, the beta hemolytic streptococci, they show complete hemolysis of the surrounding RBCs. Example includes Streptococcus pyogenes, group A beta hemolytic streptococci. And the third category is gamma hemolytic bacteria. Such streptococci show no hemolysis at all. They are also known as non-hemolytic streptococci, and they are usually not pathogenic, whereas the alpha and beta hemolytic streptococci are generally pathogenic, whereas the gamma hemolytic streptococci are not pathogenic. Example is enterococcus. I hope this presentation was beneficial to you. Thank you, and subscribe our channel for more such contents.